Hey future doctors! Welcome to Science Coat NEET, not just a channel, but your trusted partner in this journey to NEET 2025. Biology feels overwhelming sometimes, right? Especially topics like DNA replication, polymerases, helicases, Okazaki fragments. It all sounds too much. But today, let's change that. In the next 5 minutes, we'll simplify one of the most feared yet most loved NEET questions. So you can not just understand it, but remember it for life. If you're dreaming of that 650 plus score, don't just watch, feel it, revise it, live it. Subscribe, like, and share, and bring your study squad along. Because here at Science Coat NEET, we crack NEET, one concept at a time, together. Ready to own this topic? Let's begin. Hey, please welcome our respected professors. Please, sir and ma'am, let's start.
Welcome back to the deep dive. Today we're diving into something, well, fundamental to life itself. DNA replication. How cells copied their genetic blueprint.
That's right. We've got some great material outlining this, and our aim is really to unpack how this incredibly precise process works. It's quite a feat of molecular machinery.
Absolutely. When you think about it, every time a cell divides, this has to happen perfectly. So let's zero in on the core idea first. DNA replication is about duplicating DNA before cell division.
Exactly. And a really key concept here is that it's semi-conservative.
By semi-conservative, okay, what's the significance of that? Why is that description important?
Well, it means that when you make a new DNA double helix, it's not entirely new. Instead, each new molecule keeps one of the original strands, the parental strand, and gets one brand new strand synthesized alongside it.
I see. So it's like using one half of the original as a template.
Precisely. You preserve one original strand in each copy. It's crucial for stability, for ensuring the genetic information is passed down accurately.
Okay, that makes sense. One old, one new. So how does this whole copying process actually kick off? Where does it start?
It starts at very specific locations on the DNA called origins of replication. You can think of them as like designated starting blocks.
Right. And then?
Then an enzyme called Helicase gets involved. Its job is basically to unwind the DNA double helix. It breaks the hydrogen bonds holding the two strands together.
Like unzipping a zipper.
Exactly like unzipping it. This creates what's called a replication fork, that Y shape, where the DNA is actively separating.
But wouldn't the strands just want to snap back together?
Good point. They would. That's why other proteins, called single-strand binding proteins, SSBs for short, quickly bind to the separated strands. They kind of hold them open and stabilize them.
Okay, so helicase unwinds, SSBs stabilize. But doesn't unwinding a helix cause, like, tension further down the chain, like twisting a rope?
It absolutely does, and that's where another enzyme, toisomerase, comes in. In bacteria, there's a specific type called DNA gyrase. Its role is to relieve that twisting stress, preventing the DNA from getting tangled up.
Makes sense. Sort of managing the strain. What happens next?
So once the strands are separated and stabilized, another enzyme called Primase lays down short little stretches of RNA called primers.
RNA, not DNA.
Yeah, RNA primers. Because the main DNA building enzyme, DNA polymerase III, needs a starting point. It can't just begin synthesizing from scratch on a bare strand. The primer gives it a place to attach.
Got it. Like a little flag saying, start building here.
That's a good way to put it. So DNA polymerase III latches onto that primer and starts adding new DNA nucleotides, extending the new strand. But here's a key thing. It can only add nucleotides in one specific chemical direction, the 5' to 3' direction, always.
Okay, 5' to 3'. Does that cause any issues?
It does for one of the strands, because the two DNA strands run in opposite directions. So one strand, called the leading strand, can be synthesized continuously, just following the replication fork as it opens up. Smooth sailing, 5 to 3 fit.
But the other one?
The other one, the lagging strand, is pointing the wrong way relative to the fork's movement. So to synthesize it also in the 5 to 3' direction, it has to be made discontinuously in short little bursts.
Okay. Pieces instead of one long strand.
Exactly. These pieces are called Okazaki fragments. It synthesizes a fragment, then hops back as the fork opens more and synthesizes another fragment.
So you end up with all these fragments on the lagging strand. How do they get joined up into a complete strand? And what about those RNA primers?
Right. So another enzyme, DNA polymerase, comes in. Its job is to remove the RNA primers and replace them with the correct DNA nucleotides.
Okay, primer's gone, replaced with DNA. But there are still gaps between the fragments.
Precisely. And that's the final step in joining. An enzyme called DNA Ligase acts like molecular glue. It seals the nicks or gaps between the Okazaki fragments and where the primers were replaced.
Creating one continuous, unbroken strand.
Yeah.
Wow. That's quite the coordinated dance of enzymes.
It really is. Helicase, SSBs, topoisomerase, primus, polymerase III, polymerase, ligase, all working together.
And the accuracy is supposed to be phenomenal, right? How does the cell make sure mistakes aren't constantly being made?
Well, DNA polymerase III itself has a proofreading function. It's kind of amazing. As it adds nucleotides, it can sense if it's put the wrong one in. If it detects a mismatch, it can actually back up, remove the wrong nucleotide that's called its 3'5' exonuclease activity and put the right one in.
So it double checks its own work.
Essentially, yes. This proofreading makes the process incredibly accurate. We're talking about maybe 1 error for every billion nucleotides copied. It's crucial for preventing mutations.
1 in a billion. That's just astounding precision. And just to quickly recap some key points, this whole semi-conservative idea was proven by the Michelson-Stahl experiment, right?
That's the classic experiment, yes, using heavy nitrogen isotopes. And all this replication machinery is active during a specific part of the cell cycle, the S phase, which stands for synthesis phase.
Right. And those enzymes, helicase unwinds, primus primes, pol third builds, pol 1 replaces primers, ligase seals, and topoisomerase relieves stress.
Got it. And remember, the Okazaki fragments are the hallmark of that lagging strand synthesis, all dictated by that fundamental 5-to-3 directionality.
This has been a fantastic breakdown of such a complex process. It's mind-boggling to think this is happening constantly inside us.
It really is. And thinking about that semi-conservative nature, it ensures that each daughter cell gets one of the original strands, maintaining that direct link back through generations. It's a beautiful system for fidelity.
And the teamwork of all those enzymes.
Yeah.
It makes you think, what if just one of them wasn't working correctly? The whole system could break down.
Exactly. It highlights the complexity and the robustness, but also the potential fragility of these core life processes.
For anyone listening who wants to dig deeper, maybe check out the details of that Michelson-Stahl experiment, or look into how replication differs slightly between simple bacteria and more complex cells like ours. It leaves you wondering, doesn't it? What other intricate mechanisms are ensuring life continues just beneath the surface? Thanks for walking us through this today.
Thank you so much, sir and ma'am. That was a powerful and crystal clear explanation, perfect for NEET 2025 revision. Hey dear students and listeners, how did you find this episode? Drop your thoughts, doubts, or favorite part in the comments section below. I'd love to hear from you. If you found this helpful, don't forget to subscribe to Science Coat NEET, your trusted channel for daily NEET 2025 tips, tricks, and complete concept clarity in biology, physics, and chemistry. Hit the bell icon so you never miss an update, because the next season of high-yield NEET content is coming very soon, and it's going to be fire. Let's crack NEET, one concept at a time, together.